we're going to start with pillar 3 this afternoon. If you're still eating, feel free to take your food, bring it in here, eat up, take your time, have coffee. But we have a really great talk ahead of us, an amazing speaker, who's Dr. Alvin Huang, who's coming to us from Brown University. He's a translational assistant professor of molecular biology, cell biology, and biochemistry at Brown. And he's going to be talking to us about new insights into Angelman syndrome, exploring UBE3A's diverse role in brain development and health. Dr. Huang.
Yes. Right. Wow. Well, thank you for having me here. So we have learning all the beautiful genetics behind the Angelman syndrome, and we have been so excited about the progress in gene therapy, aiming at restoring UBE3A. But for these pillar 3 sessions, I want to shift our focus on understanding what this crucial gene actually does. Because by digging into the function of UBE3A, we can uncover new ways to counteract its loss in Angelman syndrome and potentially leading to effective therapies. So first, we will explore what UBE3A is. So specifically, this important enzyme called ubiquitin protein ligase E3A, and why it's vital for maintaining protein balance in our cells. And next, we'll discuss how we might restore the protein balance in neurons by addressing this loss of UBE3A function, which could offer a promising therapeutic approach. And finally, we will delve into UBE3A's surprising role in supporting helper cells called oligodendrocytes in the brain, revealing how this unexpected function could lead to new therapies for Angelman syndrome. So what exactly is UBE3A? So to break it down, think of ubiquitin as a small label or tag that marks unwanted protein in our cells for removal. So much like a recycle me stickers you might put on an old appliance. And a ligase is an enzyme that ligate or join like two molecules together. So in this case, it attaches ubiquitin tags to proteins. So therefore, UBE3A is part of the enzyme system that takes specific proteins with ubiquitin and signaling that they may be removed and recycled by the cell's waste management system. So UBE3A is super important for this ubiquitin proteasome system, or UPS as we call it. And imagine that this UPS as the cell's recycling centers that manages and recycle unwanted proteins. So within these systems, there is a tagging team composed of 3 types of enzymes. So E1, which is enzyme type 1, are like the activators. So there are only about 2 types of them. And E2 enzymes, they act as carriers with around 30 to 40 types. And then the E3 enzymes, numbering approximate at 800 types, including UBE3A, are the takers. They decide which proteins get tagged with ubiquitin for recycling. So once the proteins are tagged, they were sent to the proteasome or the cell's shredder, which breaks them down into reusable pieces. And UBE3A is crucial because without it, some unwanted proteins don't get tagged for removal. So they start accumulating and potentially causing problems in the cell. And this is especially important in the brain, where the buildup of certain proteins can disrupt normal cell function. And beyond the UPS system, our cells actually have another method for getting rid of unwanted materials called autophagy, which you can think of as the cell's cleanup crew. So autophagy handles large unwanted items, like big pieces of furniture that regular recycling can't process. So a special structure called autophagosomes acts like a garbage truck collecting these large items, and then the autophagosome then deliver the waste to the lysosome, so the cell's waste processing plant, where it's broken down. And strikingly, we found that UBE3A plays a role here too, as it helps control the number of garbage trucks or autophagosomes kind of on the streets and preventing the over cleanup. So when UB3A is present, it ensures that only the necessary amount of material is removed. Without UB3A, too many garbage trucks are active, removing not just waste, but also important items for the cell or the cell still needs. And this can lead to the loss of essential proteins, which is problematic for cell health. And so the overactive cleanup process here has very significant impacts because our ability to learn and remember depends upon rapid and precise communication between neurons, which connect at points called synapses. And the right balance of certain proteins at these synapses is very crucial for effective communication. So without UB3A, the overactive autophagy leads to the excessive removal of essential proteins at synapses. And this imbalance disrupts communication between neurons and contributing to the learning challenges often experienced by individuals with Angelman syndrome. So understanding this critical role of UB3A in regulating autophagy and maintaining protein balance brings us to even important questions. So how can we address the loss of UB3A function and then to restore the protein balance in neurons as a potential therapy? So if we can find a way to compensate for the missing UB3A, we might be able to counteract this overactive autophagy process and relieve some of the symptoms associated with Angelman syndrome. So in our research, we have identified a key player called LC3B, which acts like the engine of the autophagosome or the garbage truck in our earlier analogs. And LC3B is critical for driving the autophagy process and needs to be carefully regulated to maintain the right level of cellular cleanup. UB3A normally helps control LC3 labels by taking it for degradation when necessary. Without UB3A, LC3B accumulate, leading to overactive autophagy. The garbage trucks go into overdrive, removing too many important proteins. So using advanced AI techniques, which just won the Nobel Prize this year, indeed. So we have been able to visualize exactly how UB3A and LC3B interact. So this detailed understanding help us figure out the precise mechanism by which UB3A regulates LC3B, opening the door to potential interventions. With this new knowledge, we wonder if there's anything we can do to restore the balance of LC3B and autophagy to compensate for the loss of UB3A. So if we can find a way to regulate LC3B levels without UB3A, we might be able to bring autophagy back to normal levels, preventing the excessive removal of important proteins and improving neuron functions. So to tackle these challenges, we search for a compound that can step in for UB3A, and from a large library of compounds, we identify one, calling it compound D5. So D5 works by binding to LC3B at the same spot where UB3A would normally interact. So we believe that D5 can mimic UB3A's effect on LC3B, effectively restoring the regulation of autophagy. By using D5, we might reduce autophagy to normal levels in cells lacking UB3A, which could help prevent the loss of essential proteins and improve cell function. So our preliminary results are very exciting to us. In our laboratory studies, the treatment with compound 5 reduced the overactive autophagy in cells without UB3A, and then this reduction helped restore normal synaptic function, which is essential for learning. And we are now moving forward to test D5 in animal models of Angelman syndrome to see if it can improve the brain health in a living organism. And for us, this really matters because if successful, D5 can become a potential therapy to compensate for the loss of UB3A. And additionally, similar compounds are already being tested in clinical trials for other conditions and suggesting that they may be safe for use in humans. So our ultimate goal here is to assess whether D5 would hold the potential to be further developed as a new therapy. All right, now we want to switch the gear a little bit because as we dive deeper into understanding UB3A, we've found that it does more than just help balance proteins in our cells. It also plays surprising roles, especially in helping the spatial support cells in the brain called oligodendrocytes, which are a type of glial cells. And to explain how these glial cells work, so let's first think about how neurons, our brain's nerve cells, communicate with each other. So most of us know that when we learn something or notice something around us, this information is passed through our brain like an electricity traveling through wires. So let's just imagine that the brain is a sophisticated electric circuit board. So in this circuit board, the nerve fibers of neurons act like wires that allow electricity, or in our case, information to travel from one point to another through connections, which is the synapses. But just like electric circuit board, there isn't only about the wires. Our brains have other crucial components. So, on a circuit board, we have resistors, we have capacitors, and then other parts, they can control the fine-tuning of the flow of the electricity, making sure everything works efficiently. And similarly, in our brains, glial cells like oligodendrocytes are essential helpers. So they wrap around nerve fibers with a protective layers called myelin, so which acts like insulations on the electric wires. So this myelin sheet is kind of like an electric tap that helps information travel faster, more smoothly, and without this helper cell, our brain's communicating system wouldn't work properly. And then these well-wrapped nerve fibers create what we call white matter in the brain, which we can actually see on a brain MRI scan as the bright subway system of the brain lying beneath the gray matters, the less insulated outer layer of the brains that handles all the processing. And interestingly, this myelination process by oligodendrocytes is the very final touch to building our brains. Even so, our neurons, they are lined up and ready for action early on a baby's brain inside the womb. This myelination really takes its time. So our brains keep fine-tuning this myelination well into our use. And in fact, it isn't until we are in our mid-20s that this process is fully complete. So that's why we keep getting smarter and better at solving problems as we grow from teenagers into young adults. But why is this important for Angelman syndrome? Well, so multiple studies, including several amazing groups within our FAST community, they have been using brain MRI scan and have shown that young people with Angelman syndrome, so ranging from babies, children, teenagers, or young adults, experience a slowdown in the way their brain become insulated with myelin. So this is like brain's wiring taking longer to get its protective coating, which obviously can affect learning and behaviors. So the good news is that some researchers find out they seem to catch up when they become adults. But since the period of childhood to young adulthood is so crucial for developing intellectually, so the delay can make learning tougher for the kids during those years. And importantly, it turned out that UB3A is also expressed in oligodendrocytes in human brains. And this report has made our research team curious about whether UB3A has a job in helping oligodendrocytes grow and work correctly in our brains. So our amazing postdoc researcher, Dr. Xin Yang, then decided to study this problem using the human cells that you've heard so many times about, the iPSC cells, to gain more sort of relevance to the human condition. So this technique, we think is really cool, is that these iPSCs, they carry all exactly the same gene as the people who donated, but can also be turn into any brain cells of our choice. And we have discovered that UB3A is active during the early stage when oligodendrocytes are developing. And we are also thrilled to collaborate with Dr. Yonghui Zhang at Yale and access to this valuable resource, which is a collection of iPS cells donated by Angelman syndrome patients facilitated by FAST, that you will learn more details later. And in the case of Angelman syndrome, we find that gene mutations in our donor iPSCs can deactivate UB3A, and we think this is why young people with Angelman syndrome experience slower myelination process. And our team is thrilled about this, also because we know that we found just another new way to possibly help fix brain cells issues that affect how the brain works. So we have been using such stem cell methods to screen a collections of handful of drugs in a very unbiased manner, hopefully to speed up our search for a potential therapy. And so please don't worry about the exact data showing here, but I just want to shows that our data are so encouraging to us is that we find out like a field of drug compounds we identify indeed can restore the normal growth to this struggling oligodendrocyte. And interesting that this effective drug interact with the oligodendrocyte in the ways that we kind of didn't expect, and then we are amazed to see the significant improvement in the mouse models. And so these stuffs, they are not yet published, but if you have any questions, feel free to reach out. I will be very happy to provide the details, and big shout out to my wonderful research team as well as FAST for the generous support here. Okay.
We have a couple minutes for questions if there are any. Are there any very, very exciting questions, or can it wait for the panel? Let's see. Any hands?
Yes.
Great work. That's a fascinating idea of using a small molecule to mimic UB3A. Have you guys noticed any off targets with this?
Oh, so we identify this compound just because the very exact molecular mechanism that we think this compound can sort of step in the event of UB3A loss. So the specific target we are looking at is a protein called LOC3B, and in the analog I gave, it's kind of the engine of the garbage truck. And then, a very crucial role for UB3A to play. So hopefully, this could be a way to compensate the loss of UB3A. But the caveat is that, as we learn more about UB3A, I think, to me personally, I just got more amazed by, how this small molecule can function in the cells. So I believe that this compound, together with other excitement brought up within our FAST community, I mean, in the future they probably would have the potential to work together and really deliver, I think, the kind of therapies we all are expecting.
I'll ask a quick question.
Yes.
You said that some of these compounds are in clinical trials or approaching clinical trials for other indications.
Yeah.
Do we know, since you don't have in vivo work yet, do you know if it crosses the blood-brain barrier and how these compounds are being delivered?
Yes. Also, some of the compounds under the clinical trials, they are specifically designed to have a better penetration into the brain. So the specific compound we are looking at, they should bear certain capability to go across the brain, based on the peptide structures we can analyze. But of course, we will first test out in the mice first, and then it's an ongoing process. And if we see some promise, we certainly will be open up for collaboration within this community to better sort of tailor this type of compounds to suit our need.
one question here.
Hi, thank you very much for the talk. It was really, really enlightening. You talked about two things. One was the dumpster truck, and then the second was about the grey matter and white matter growth.
Yes.
There was a reference to, need to suck in the belly here. There was a reference to DMSO on the slide, and that's something that's been tried by some parents.
Well, thank you for catching that up. So, I didn't really have time to go into the data, which is very exciting to me, so I decided to bring it here. So DMSO indeed is a vehicle, it functions like a control, to make sure that we didn't mess up the oligodendrocyte functions when we are treating cells with, a collection of compounds we choose. Yeah.
Okay, so that's not a potential therapeutic as part of this.
Yeah, it just shows, shows that what would be the sort of normal or baseline conditions of our oligodendrocyte when they don't have UB3A.
Okay, fair. And just one follow-up question.
Yes.
Is there any natural occurring products or things that could be used to help with the grey matter growth? When I saw some of the studies on grey matter growth, I was really disappointed because it was like, oh no, there's like, this is going to be such a big thing to overcome. So I was just wondering if there's anything natural that parents could do to help with grey matter growth in advance of these therapeutics.
Yes. If this probably will be a little bit sort of oversimplified, but normally, the scientists would view gray matter as where most of the neurons are located, and white matter as the parts that the glia or supporting cells are located. So as many of us know that neurons, they have a very limited capacity to grow once they are born. So you are right in the ways that when we have loss in gray matters across all the brain disorder spectrums, it's actually very difficult to grow them back. But there are, in other disease contexts that, if we have to dealing with a brain that has a much limited of this regeneration power, there are certain sort of regeneration therapy can help to restore gray matter to some extent. But I don't think, overall for Angelman syndrome, the brain or neurological conditions we are mainly dealing with, we don't really, have this big issue of loss of gray matters.
Okay, great. Thank you very much.
Sorry, I just have a quick question too. So DMSO, there is some research into how it helps with cerebral palsy. That's an anti-inflammatory. So how can it be used as a control if it actually crosses the blood-brain barrier and has an impact?
Yes, so DMSO is a very interesting chemical, I have to say. So in the laboratory settings, we mainly using as a solvent, and because it can help to dissolve a lot of compounds, as Alison mentioned, they may not readily go across the blood-brain barrier, reach the brain, and so we can use it kind of as a vehicle to do that. And the reasons we include DMSO here, because we use DMSO as a solvent for all the drugs we are testing. So in some ways that the DMSO condition for us is kind of a blank, empty vehicle versus, the condition that we have a vehicle plus the drug. And this is, I think, also unique for the neuroscientists, is that we constantly really have to deal with these issues about the delivery because, neurons, they are just notorious to be reached. But you're right, DMSO also has been, has this like a wide range because, they have this ability can kind of stabilize the lipid component in the brains. But for us, I think we can do something much better than the DMSO. So for us, we treat it as a vehicle here.
Thank you.
Okay, we'll reserve any more questions for the panel. Thank you so much.
Thank you.